The following presentation is off the research paper Menin mediates epigenetic regulation via histone H3 lysine 9 methylation.
This paper has been chosen from the National Centre for Biotechnology Information website. It has been selected from 94,054 papers, and this is the link to the paper.
We have chosen this paper because we're interested in finding out more about Menin tumors and because it is relevant to the topics we have studied in molecular biology. It relates to histones, methylation, gene expression, transcription, proteins, and repressors.
This research paper investigates whether menin functions as a transcriptional activator to mediate histone methylation and a repressor. This is the abstract taken from the article summarizing the experiment.
These are the key terms we believe are relevant to this experiment.
Menin is a tumor suppressor protein encoded by the ME1 gene. Menin has a role in multiple biological pathways, including gene transcription, cell cycle control, and genome stability. Menin can lose its function due to either missense or nonsense mutations along the entire gene. SUV39H1 is a gene, a member of the suppressor of the variegation 39 homolog family, and encodes a protein with a chromodomain and a C-terminal set domain. This nuclear protein moves to the centromeres during mitosis and functions as a histone methyltransferase, methylating lysine 9 of histone H3. Overall, it plays a vital role in heterochromatin organization, chromosome segregation, and mitotic progression.
Immunoprecipitation is a technique in which an antigen is isolated by binding to a specific antibody attached to a sedimental matrix. It is also used to analyze protein fractions separated by other biochemical techniques such as gel filtration or density gradient sedimentation. The source of antigen for immunoprecipitation can be unlabeled cells or tissues, metabolically or intrinsically labeled cells, or in vitro translated proteins. Methodology. The scientists cultured 293 T cells in Dobesco's modified Eagles medium. The 293 T cells were transfected with transfection reagents. For Western blotting, 293 T cells were harvested 24 hours after transfection and lysed with lysis buffer. The obtained lysates were immunoprecipitated with appropriate antibodies. The immunoprecipitates were washed 4 times with IP buffer and with HMT buffer. Samples boiled for 5 minutes and the proteins were then fractionated. The deletion mutations of menin were synthesized and labeled with S-methoimine using in vitro kit. Menin plus and menin minus cells were transfected with control menin for 24 hours. Quantitative real-time PCR was performed with specific primers for GBX2 and GAPDH with qPCR kit. Arrays were scanned with an Illumina bead array reader, and array data export processing and analysis was performed using Illumina bead studio. Chromatin immunoprecipitation was performed.
The results of this experiment show the menin interacts with SUV39H1 and this ability might be involved in its tumor suppressor function. SUV39H1 interacts domain is mapped on a distinct region of menin by comparing 2 regions mapped for SUV39H1 interaction using comparative protein structure modeling with threading method. Menin and SUV39H1 have common targets for gene regulation. 649 genes were identified as overlapping with the genes whose expression was elevated by the lack of menin, suggesting that a subnet of genes are co-regulated by menin and SUV39H1. Epigenetic regulation mediated by menin depends on the context. The regulation of IGF-BP2 is mediated by menin via HDAC. Menin recruits SUV39H1 to promote chromatin remodeling at the GBX2 locus. When the promoter at the third end region of GBX2 were compared, changes in histone modification were prominent in the promoter that menin occupied.
The following figure is taken from the research paper and it demonstrates that menin and SUV39H1 contribute to GBX2 repression.
Menin functions as a tumor suppressor in endocrine tissues and an onogenic cofactor in blood cells by playing within complex protein interaction network. One of the key interacting partners of menin is MLL H3K4 HMT menin, and MLL mediate transcriptional activation of COK inhibitor genes. Menin integrates with SUV39H1 for gene repression and together target GBX2.
To conclude this experiment, we have to ask ourselves, what is the underlying mechanism that allows menin to function as a transcriptional activator or repressor? Interactions with SUV39H1 can be activated or repressed by the phosphorylation on thyroid residue of parafibromin. Parafibromin coordinates transcriptional events and chromatin remodeling, which is important for gene expression. The following figure illustrates a model for a dual role of menin in gene regulation during tumorigenic pathways. Menin may function as a transcriptional activator or repressor in a context-dependent manner by selective mediation of chromatin remodeling, which provides an efficient mechanism for the regulation of gene expression and cell tumorigenesis.
The results and conclusions obtained from this study provide the opportunity for a potential future study to identify interacting partners and their critical targets, as well as a work to disintegrate the dynamic nature of the interacting network to get a deeper understanding of menin's role in gene expression. 44. Is that acceptable?